Welcome to Ask the Expert. Today we have with us Chenqu Suo. She is a clinical PhD student supervised by Dr. Sarah Teichmann at the Wellcome Institute in England, and she's obtained her medical degree from the University of Cambridge and was a pediatrician in training before joining the lab in 2019. Her research interests lie in human immunology, in particular immune system development, in vitro cell engineering, and implications in understanding and managing diseases. In her recent publication in Science, congratulations, that's fantastic, she spearheaded with another PhD from the Teichmann lab, Emma Dan, on reconstructing the developing human immune system across multiple tissues using single cell technologies. And that is no small feat. So welcome, Chenqu. Thank you very much for joining us. And yeah, would you like to share some of your work with us?
Sure. Yeah. Thank you very much, Monica. I will start sharing the screen now. Could you see it now?
Perfectly.
Okay, great. So thank you very much for inviting me here for the talk to talk to sort of the research community of type 1 diabetes. And I really have the pleasure to share our recent work here on this platform. So my talk today will be on mapping human immunity one cell at a time. And I'm talking not only about our own project, but also on another piece of work from our lab that's also published in Science in May. Right. So to start with, probably most people have heard about the Human Genome Project, whereby the main aim is to sequence and map the all of the genes in our genome in the human cells. However, although this is a sort of great initiative, but what it doesn't tell us is that it doesn't encode the multitude of different cell types within our body, although we have the almost same DNA information across the different cell types, but of course they look different and they function differently. So how do we solve that problem? And here my supervisor, Sarah Teichmann, worked together with Aviv Roger from Broad Institute and started the initiative of Human Cell Atlas in 2016. And what Human Cell Atlas is about is really that it's to create a comprehensive reference map of the different kinds of cell types across the whole human body. And this would include in both healthy condition as well as in disease. And really this has been sort of a rapidly evolving global effort, and the community now really spreads sort of 79 countries and including over 2,000 members from over 1,000 institutes as well. So if you are interested, please have a look on our website, and you're welcome to join the community as well. But of course, that almost sort of the majority of the new discoveries are often preceded by new technologies. And what's the technology that's underpinning the great discoveries of different cell types here? And the key technology that we use is single cell genomics. And I'm here just going to explain a little bit for people who haven't heard, although I believe that most of you might have already heard or even used this technology. So the majority of the time that we use single cell RNA sequencing, and what it is that if we take a piece of tissue, for example, in this case, the heart, and digest into single cell suspensions, then the technology allows us to obtain the RNA sequencing of all the genes within a single cell, each single cell. So
that's particularly important in the islet, right? Because it's made of several very different types of cells.
Of course, yes. So definitely in the pancreatic islets, that has different cellular heterogeneities. And of course, that sort of the multitude of information and sort of we are looking at each cell with a magnifying glass. So this zooming in action helped us to tease apart more heterogeneity within the cell types. So this is sort of the resolution revolution that's underpinning Human Cell Atlas project. And on top of that,
resolution revolution. It's fantastic.
Yeah. Thank you. And on top of that, but what sort of information is being lost in the single cell RNA sequencing is that, as I told you, we have to digest tissue into single cell suspensions. So where the cells are coming from, so that spatial information is lost. And to complement that, we also have the spatial transcriptomics. So from the same tissue, we could cut it and put it onto a slide. And then on that slide, the tissue is put into different grids, as you could see here. And for each spot of that grid, we then could do almost like a sort of small mini bulk RNA sequencing of that spot. And with that information, the intention is then to deconvolve into different cell types. So we know exactly which cell types are located in the tissue. So to put them really back to their tissue context. So of course, we need sort of the computational tool that links these two technologies together. And so Oma, also from Wellcome Sanger Institute, so our adjacent lab, has developed this computational tool called Cell to Location, which is again published this year in Nature Biotechnology. And what it does is that it uses single cell RNA sequencing as the reference data to deconvolve the spatial transcriptomics, so that it allows us to identify at each spot the abundance, so what cell types are present there and the abundance of that cell type, as you could see from the illustration here. So the advantage and also the power of this tool is that it can take different reference data, so single cell or single nuclear RNA sequencing, and it can be used in different technologies of spatial transcriptomics as well.
Can I ask you, can this technology also be used to ascertain the microbiome as well? That might be a like, you know, basic question, but.
So I guess, I mean, personally, I haven't studied microbiome myself, but I guess if you could put, let's say like a piece of gut tissue on the slide, and if we could preserve the microbiome that's on top of the gut epithelium well, then I think that's possible as well, yes.
Great, thank you.
And so what do we do in our lab, in the Teichmann lab, is that, so we've really started atlasing the different organs since 2018. So we first started with sort of each individual organs first, as you could see, sort of maternal fetal interface, the gut, heart, muscle, lung. But what's really sort of new, but also an important step in our two recent publications, is that we are beginning to really assemble the atlases across different tissues and across different organs, so that we are obtaining like a comprehensive map of the human across different tissues. And this is really towards sort of an important step towards a completed draft of the human cell atlas. And here, so the publication that I co-led with Emma is on the developing immune organs, and the publication from Cecilia and Chuan from our lab is on cross-tissue immunity in adults. So I'll focus on these two papers today. And I will use the, so our papers, the integrated developing immunity here, to illustrate what sort of important biological insight you could obtain using the different technologies that I've about. And I'll use Cecilia's paper, that cross-tissue adult immunity, to highlight the computational tool that our lab has built to facilitate the sort of other users of the same technologies to have an easy access of the technology. So starting with the developing immunity here, again, why do we want to do this is really that so far we have done analysis of each single organs, but we really want to put these pieces together to assemble the full body atlas. And this is particularly important in the immune system, because the immune system is really a distributed network. You can find the immune system almost in all of the organs present in the human body. And so the immune cells first start to develop in the primary hematopoietic organ, so which I meant the organ that's responsible for generating the blood cells and the immune cells. And in adulthood, that's the bone marrow. And then the developed immune cells then start to migrate to populate the lymphoid organs, such as so the thymus, lymph nodes, tonsil, spleen. And the thymus is particularly important because T cells really complete their development and maturation there. And also the immune cells migrate all around the body to populate the non-lymphoid peripheral organs as well. And it's really the same immune cell types that we see throughout the whole distributed network. And so the situation is very similar in prenatal development. And the only difference is that in the primary hematopoietic organs have a constant shift in prenatal development, whereby it starts from the extraembryonic yolk sac, which then the hematopoietic role is taken over by the fetal liver, and then is taken over by the bone marrow, which then persists to become the dominant hematopoietic organ in adulthood. But apart from that, the whole sort of development of immune cells and the how they populate the rest of the organs is very similar to in adulthood. And how we are looking at this distributed network is that so we have collated different databases from different data sets from the public database, and also generated some new data ourselves as well, so that we have the single cell RNA sequencing information from all of these 9 different organs, so that it has a good representation of the hematopoietic organs, the lymphoid organs, and the non-lymphoid peripheral organs. And on top of that, we have also done paired single cell VDJ sequencing. So we have a deeper dive into the antigen receptors of B and T cells, and this is perfect to study the immunity really. And on top of that, in addition, we have also generated spatial transcriptomics. Like I said, this is to place the cells we see back to their tissue context. And we have also done some validation work using in vitro differentiation, etc., so that overall this creates a comprehensive developing immunity atlas. And this comprehensive atlas has really been very useful in generating new biological insights. And so the boxes highlight some of the biological insights that we have made in our manuscript. And due to time limit, I'm just going to highlight one of them, which is the system-wide blood and immune cell development. So this is really quite surprising. So what I've told you just now is that what we know from textbook and from previous literature is that blood and immune cells really develop from the primary hematopoietic organs, which in this case is the yolk sac, the fetal liver, and the bone marrow. However, what we have observed in the single cell data is that we could see the immune cell progenitors almost everywhere in almost all of the organs that we have sampled. And in particular on B cell development. So here I'm going to show you some of the results we have. So this is a UMAP of all the immune cells. So each tiny dot that you can see, it represents a cell. And here the colors are just highlighting the progenitors that we were interested in of different lineages. And the different colors highlight the different cell types that we have annotated as well. Because for each tiny dot of this, which is a single cell, we have a multitude of information and we know exactly from which organ this is coming from. So when we looked at the B progenitors, as you could see that the B progenitors start from the pre-pro B cells, the pro B cells, late pro B cells, and then large and small pre-B cells, which is the whole transition of B cell development that has been described in literature. But when we looked at the cells from different organs, we could also find these B progenitors in all of the organs that we have sampled. So not just the primary hematopoietic organs. So as you could see here in the plots underneath, so each yellow dot represents a cell from this organ with name on top. And you can see that we can find these progenitors really throughout the different stages in their development as well in all of the organs. And this is truly interesting because what it shows us is that the B cell development happens in almost all of the organs that we have sampled. And then we looked at the spatial transcriptomics to see, so where about do we find them? So on the left panel, shows you the results from the VCM, which is the spatial transcriptomic data. So for each organ, the small picture shows the H&E, highlighting the structure of that organ. And then the right picture, the color scale shows the abundance of the B progenitors predicted. And so we could really see B progenitors being in all of these different organs. For example, in the prenatal gut, developing gut, we could locate these B progenitors in the submucosal region. And we have also used single molecule fluorescence in situ hybridization to further validate that truly we could identify these progenitors in situ in the organ. And here, so each dark purple spot represents a nucleus, and then the different colors are represent the RNA expression of the different genes. So the green, blue, and yellow are the genes that are only expressed during B cell development. So this is during the B cell receptor recombination process. So as you could see that these highlights the B progenitors we could see from the developing gut. And then the CDH5 is a gene that's specifically expressed by endothelial cells. So it really highlights the blood vessels. So here you can see B progenitors that are associated with blood vessels, which are highlighted by the gray arrows. But also we could see the B progenitors being far away from the blood vessels, highlighted by the golden arrows as well. So this means that we're not just observing the progenitors because they are carried to the tissue from circulation, but actually they could be found inside the tissue away from the blood vessels, which means that the development of B cell happens in situ there.
They develop in residence as well as, coming from the blood system.
Exactly. And also together with what I showed you earlier, you could see through different stages of the development. This means that the development happens there in that tissue as well.
That's great.
Yes. And then, so this is just a 3D rendering of the SM fish picture that I've just shown you, just to further highlight that, if you focus on this cell and this cell, that they are truly away from blood vessels here.
Beautiful imagery.
Thank you. And then, so of course, so what's the importance of this, right? So this is surprising because we find B cell development everywhere, but what are the supporting cells that help in the development? And this is important because we want to create the same development in a dish as well. So with cell engineering, etc. So what we did is we went back to the spatial transcriptomic data and then looked at, so what are the cell types that co-localized with these B cell progenitors in the different tissues? And we have identified, so ILC3, specific subset of macrophages, some natural killer cells, and also predicted the interactions between these immune cells with the B cell progenitors. So again, this is quite surprising because for people who have been looking at stroma that's supporting B cell development, I mean, the majority of the studies were done in mice, but really people have only been looking at the, the fibroblasts, the endothelial cells, so the non-immune population. But what we might be suggesting here is that it could potentially be the other immune cells that might be contributing to the B cell development here. And that might be why that, it's such a universal feature. You could find it almost in all of the developing organs. So of course that this can be an alternative niche to the bona fide sort of bone marrow stroma, and it has important implications on how we engineer B cells in the dish.
Oh, yes, it does.
Yes.
I mean, it really does. Yeah, it's a real game changer, right?
Yeah, definitely. And actually, so we are following on from here and working with another lab as well to see how we could take this further to help engineering B cells in the dish. So I think, I'm just going to highlight this one particular biological insight from our developing immunity paper. And like I said, this work was co-led with Emma Dunn and another computational PhD in our lab, and we're both supervised by Dr. Sarah Teichmann. And this is collaboration with Professor Mars Hannifer from Newcastle University and Mena Clatworthy from University of Cambridge. And our paper has been online in Science.
That's
and
fantastic.
then,
oh, sorry, I just wanted to ask you just quickly.
Yes, sure.
Are you, are you collaborating across, the Atlantic, basically with Genentech, with Aviv currently? Does your group do that?
Yes, well, so I'm not sure, I'm unsure whether we have any active projects, but there's a lot of communication between our lab and Aviv's lab as well.
That's great. And then are you, I mean, maybe we can leave this to the end as well, but
Sure.
Are you looking for, postdocs, medical students, things like that, or anyone in particular with some expertise in X, Y, and Z?
Right, I see. I think I would say yes, but of course that question probably I need to ask my boss, Sarah Teichmann. But Sarah is always interested in sort of recruiting people with different backgrounds. So our lab is actually filled with people from different background. Like we have people from immunology who did immunology, from bioinformatics, and from medical background like myself. This has because we not only want to do Atlas in sort of healthy conditions, right? We want to expand this into disease as well. So yeah, of course, I think anyone who's interested in our work should contact my boss and the lab.
Yeah, no, it just seems like such a rich environment and just a wonderful place to do the work. So yeah, keep going. Sorry.
Yeah, no worries. Right, okay, so then next I'm really speaking on behalf of Cecilia for their work on cross-tissue adult immunity. of course, that the starting point was very similar, that we want to assemble a comprehensive atlas across the whole body. But I might have sort of hinted towards it that one particular challenge in using single-cell RNA sequencing is actually cell annotation. And I think a lot of labs spend a lot of effort and time on annotating the cells. Yeah. Yes, because it's such a multitude of information that you get for each single cell, and the traditional methods of using, just one or two markers could help, but of course would not be the best method to use here. And really, we also need a cell type encyclopedia, right? Because different people call different cell types slightly differently, and the definition might be different across different labs as well. And what we want is to create the sort of almost homogenized reference map that everyone could use. So to start with, we have worked on how do we sort of homogenize cell type annotation and also help people and automate the process. So here the computational tool that we've created is called Cell Typist, and what it is that it creates an automated cell type annotation process. And so Cecilia and Chuan from our lab, together with a few others from the collaborating labs, have really spent a lot of effort going through the public data sets, and they've curated and homogenized the labels, i.e. the cell annotations from these public data sets. And then they've used these data sets as a training model to train the cell typist, which basically is based on a machine learning algorithm. And with your own input single cell RNA sequencing, then what cell typists could do is output for each single cell the predicted cell type annotation. And of course that they have also assembled these adult immune cell atlas from 12 different donors, and they have used their own data that they've created to try this out as a proof of concept that it does work, and it works very well. So cell type is, like I said, it's a machine learning model. It's mainly based on logistic regression with stochastic gradient descent, and it's a tool for precise and rapid cell annotation. So typically that when we run on a data set, it really takes a couple of minutes and less than half an hour to finish the automated annotation process.
That's
and
great. Less than half an hour to finish the entire automated annotation.
That's right. Yeah, it's really quick. Yes. And so far, from the public data sets, they have homogenized more than 100 immune cell types and states. And it's a model encompassing all tissues. Like I said, Cecilia have demonstrated in their paper that you could predict the same cell type coming from different organs, and it works equally well. And here, of course, this work was mainly spearheaded by Cecilia and Chuan from our lab, but this is a collaboration work with Joanne Jones and Karash from the University of Cambridge, and their manuscript is online in Science as well. So far, what we have as a lab have been able to do is that, so we have tried to assemble this human cell atlas, and also we have created computational tools to facilitate use by other people, such as generating this human cell type encyclopedia. And by using different technologies, we're able to map the tissue microenvironments, the cell migration process, and different cellular interactions, etc. And we're doing so throughout the whole developmental stages of humankind, so all the way from prenatal development to adulthood, really. And of course that we want other people to use our atlas, and what it could be used, for example, it can help to map the molecule. So if I'm interested in predicting, for example, a drug response, I could look at the genes that this drug might affect and in which cell types that this gene is expressed. And from the expression pattern, I could then predict that, so which cells that this drug might affect and in what ways as well. And like I mentioned earlier, by studying the cell development, it could act as a blueprint for engineering cells in a dish. And of course, that we want to expand this to understand disease more, for example, in type 1 diabetes research. And I know that in the type 1 diabetes research community, there have been multiple papers using single cell technology, both sort of in looking at the pancreatic islets, but I think more commonly from the blood, which is much more readily available, right? From the patients. And of course, yes. And
well, the islets are, the NPOD is a great repository, and then Canada has their network certain, but the islets are, very precious and rare because they come from donors.
Of course. Yes, indeed. And yeah, it's precious and rare, and I guess not easily available, right?
Yes.
Yes.
This system provides like a really powerful new approach, I think, of maybe getting more out of individual islets, and sort of like rare resources. So it's really fabulous. I wondered if you guys know how many type 1 diabetes researchers actually use these applications.
You mean applications of the healthy reference atlas, or the applications of the technologies?
Both. I mean, do you have scientists approaching you to gain access to the human cell atlas, or have help with some of the tools?
Yeah, I have to say I'm not personally aware from sort of type 1 diabetes. We have had sort of many queries from other diseases as well, of course, because I mean, we are not the only single cell lab, right?
Yes.
Yes. But I guess one difficulty I could imagine is that so far in the healthy reference, we haven't really mapped pancreas really, which I think should be on the to-do list.
There's a project for postdoc.
Yeah, of course, yes, definitely. But blood-wise, we have tons of healthy PBMC data out there. That it could work as a reference atlas, really.
And it's very readily available, correct?
That's right. Yes. So in the Human Cell Atlas website, you could see what data sets are available, and usually, like, there's a link that you can click onto that leads to the download of the data as well.
Fantastic. It's really great what you're building.
Thank you. Yeah, I think this is the end of my talk. Thank you very much.
Thank you so much, Chenqu. I mean, this is really, it's just a great opportunity for those in the type 1 diabetes research realm and other interdisciplinary realms who might be interested in type 1 diabetes, sort of like, I don't even want to say anecdotally, or, coming from another perspective. It might be really a powerful place to visit and see what you might find here. So thank you again. I would encourage people to reach out if they're interested in working in this space, and really appreciate your great share today. Hope you have a great rest of your evening in England.
Yes, thank you very much, Monica.